Chapter 14, Diseases of the Upper Gastrointestinal Tract, Part 1. Learning objectives. Describe the normal anatomy and physiology of the upper gastrointestinal tract. Discuss conditions and diseases of the mouth, teeth, and gums that can impact nutritional status. Understand potential nutrition interventions for diseases of the mouth, teeth, and gums. Define GERD and dysphagia, as well as their signs and symptoms. Describe the possible nutrition interventions for both GERD and dysphagia. Identify nutrition interventions to decrease nausea and vomiting. Describe peptic ulcer disease and gastroparesis. Understand the etiology, symptoms, and potential nutrition interventions of peptic ulcer disease and gastroparesis. Identify possible complications and appropriate nutrition therapy post-gastric surgery. No other system of the human body is so intimately involved in preservation of an optimal nutritional status of the gastrointestinal tract. Any pathology involving the GI tract can have a significant effect on nutritional status. A good working knowledge of normal anatomy and physiology of the GI tract is essential to understanding the pathophysiology and subsequent medical and nutrition care of GI diseases. The GI tract is often described as a long tube approximately 15 feet in length. This description, though technically accurate, minimizes the complexity of this body system. The upper GI tract is composed of the mouth, pharynx, esophagus, and stomach, while the lower GI tract includes the small and large intestine. These organs differ from one another in both anatomical structure and specialized function. Accessory or ancillary organs that contribute to the function of the GI tract includes the liver, biliary system, and pancreas. Motility is the movement of food consumed along the GI tract. Both propulsive contractions and mixing movement serve not only to move foodstuff towards sites of digestion and absorption, but to mix foods with digestive secretions and maximize potential absorption. Secretion of the GI tract includes water, electrolytes, enzymes, bile salts, and mucus. Through the process of digestion, complex molecules are converted to their simplest form. Carbohydrates are digested from their most complex forms of polysaccharides to monosaccharides like glucose, fructose, and galactose. Proteins are converted from polypeptides to single amino acids. Lipids are digested to their simplest forms, free fatty acids and cholesterols. After digestion, these basic molecules are absorbed along with water, electrolytes, vitamins, and minerals to provide essential nutrients to every cell. Contraction and motility are regulated through a complex network of communication paths involving autocrine, paracrine, and neuronal control systems. Figure 14.1, Anatomy of the digestive system, starting from the nasal passage all the way down to the anus. Table 14.1, mate table 14.1. Neurotransmitters of the intricate nervous system and their effects on gastrointestinal motility. Table 14.1 continued. The oral cavity or mouth serves as the point of entry into the digestive tract. The mouth consists of teeth, lips, tongue, and palate. The lips assist in directing food into the oral cavity. By separating the mouth and the nasal cavity, the palate, the roof of the mouth, allows for chewing, swallowing, and breathing to occur all at the same time. The tongue's primary role is moving food from the front of the mouth to the pharynx in preparation for swallowing. The tongue and lips also play a major role in speech. After food is voluntarily placed in the oral cavity, teeth begin their work of mastication or chewing. The purposes of chewing is to break down food into smaller pieces, to mix food with saliva, and to stimulate taste buds. The tongue assists by moving food in place for chewing. The primary secretion of the oral cavity is saliva. Saliva is produced in the mouth by three parts of the salivary glands: the parotid, submandibular, and sublingual. Saliva is made of water, electrolytes, and protein. After food is chewed and mixed with saliva, it is shaped into a sticky ball called bolus. The bolus is moved from the front of the oral cavity to the back, where swallowing is then initiated. Nutrition-focused physical assessments is a critical element of the overall nutrition assessment. The oral cavity in particular is an excellent site to assess for micronutrient deficiencies. Assessment of the tongue for color and inflammation can also identify physical changes associated with several vitamin and mineral deficiencies. The esophagus is a straight hollow tube approximately 25 centimeters long and 2 centimeters in diameter. The esophagus has a sphincter muscles at each end. Walls of the esophagus consist of 4 layers of tissue. The inner layer is the mucosa, which is made of stratified subcutaneous epithelial cells. The next level is submucosa, which contains secretory cells that produce mucus to facilitate movement of the bolus during swallowing. The muscle layer consists of both longitudinal and circular muscles that coordinate movement of food bolus by alternately contracting. The chief function of the esophagus is motility. Its primary task transporting of bolus of food from the oral cavity to the stomach. Sounds simple, yet several disorders of the upper GI tract actually involve derangement in this task. An individual unconsciously swallows over 600 times each day. Each swallow is composed of 4 phases. The first phase is the oral preparatory phase, where food is chewed and mixed with saliva. The second phase, or oral transit phase, consists of voluntary movement of the bolus of food from the front of the oral cavity to the back. The third phase of swallowing is known as the pharyngeal phase. The most important part of this phase is to ensure the bolus is directed into the esophagus and is prevented from entering the trachea. The final phase of swallowing is esophageal phase. The upper esophageal sphincter is located at the top of the esophagus. This sphincter, when opens, allows the bolus to enter the esophagus. Figure 14.2, layers of the digestive tract wall. Figure 14.3, peristalsis in the esophagus. The final portion of the upper GI tract is the stomach. This organ lies from left to right across the upper abdominal, directly under the diaphragm. Portions of the stomach differ by anatomy and function. Sphincters at both ends of the stomach regulate the flow of foodstuff from the esophagus to the small intestine. Major functions of the stomach include all 4 digestive processes: motility, secretion, digestion, and absorption. The anatomy of the stomach plays an important role in protecting the gastric mucosa from the constant flow of substances that are potentially toxic to its cells. Gastric motility includes filling of the stomach, storage of foodstuff, mixing the gastric juices, and finally emptying into the small intestines. When empty, the stomach's volume is about 50 milliliters, but it can be stretched to more than 1,000 milliliters. Storage occurs primarily in the body of the stomach. Rate of gastric emptying through the pyloric sphincter into the upper portion of the small intestine is controlled by the anatomical structure of the stomach. Nutrient contents of the foodstuff, the nervous system, and the influence of specific hormones. The stomach secretes approximately 1 to 3 liters of gastric juices each day. Gastric juice is composed of water, mucus, hydrochloric acid, enzymes, and electrolytes. The mucosal lining of the fundus and the body of the stomach contains gastric glands. Several different types of cells are located within the gastric glands. Extrinsic factor is a protein necessary for the absorption of vitamin B12. Control of gastric secretion is accomplished through complementary actions of the nervous and endocrine system and involves four major chemical messengers: acetylcholine, histamine, and gastrin, which stimulates gastric secretion, and somatostatin, which inhibits gastric secretion. Acetylcholine is a neurotransmitter that stimulates chief and ECL cells. Histamine, a paracrine, acts as a partial cell to increase hydrochloric acid release. Gastrin, a hormone, stimulates the chief cells as well as the ECL cells to release histamine. Somatostatin works as an inhibitory paracrine by providing negative feedback to the stimulatory pathways. Gastric secretion are produced even before food enters the stomach and serves to prepare the stomach for the ventral role in digestion. Release of gastric secretion in response to a meal is divided into the three phases: subhelic, gastric, and intestinal phases. Figure 14.4, anatomy of the stomach. Table 14.2, control of gastric secretion. Table 14.2, continued. Digestion in the stomach is both mechanical and chemical in nature. Mechanical digestion occurs as contractions of the stomach shear the foodstuff and mix the bolus of food with gastric juices. Hydrochloric acid causes proteins to unravel or denature, allowing for more efficient digestion. Additionally, hydrochloric acid converts pepsinogen to the active enzyme pepsin. Pepsin works to cleave amino acids, di or tripeptides, from inner portions of the protein and results in shorter chains of amino acids called peptons. Within the stomach, gastric lipase, gastric lipase hydrolyzes fatty acids at the third carbon position, releasing fatty acids and diglycerides. Short and medium chain fatty acids are more easily hydrolyzed than long chain fatty acids in the stomach. Carbohydrate digestion that begins in the oral cavity is reduced because saliva amylase is inactivated by hydrochloric acid. A small amount of carbohydrate digestion may continue within the inner portions of the food bolus, which are not exposed to the acid. Absorption in the stomach is limited. No nutrients and only a small amount of water are absorbed. Exceptions include alcohol and some medications. Alcohol can be absorbed in the gastric mucosa and enters the bloodstream through capillaries of the stomach. Because the presence of food in the stomach slows this process considerably, consuming food prior to or with alcohol would slow systemic effects of alcohol ingestion. Most medications are absorbed in the small intestine. In this section, general problems associated with mouth, teeth, and gums are discussed. These are of nutritional concern because most of these conditions involve problems that interfere with adequate oral intake and can lead to nutritional defects. Conditions affecting oral health include most common dental diseases, dental caries, gingivitis, and periodontal disease. Over the past several decades, the U.S. population has seen significant improvement in prevalence in most dental diseases. Even with these improvements, dental diseases remain one of the most common health problems and is the most important example of health disparity in our country. Approximately 19% of children 5 to 19 and almost 1/3 of adults in the U.S. have untreated dental caries. The prevalence of untreated dental caries varies with race, ethnicity, and is highest among those living in poverty. Estimates range from 20 to 40% of Americans do not regularly have access to dental care. Dental caries or tooth decay is destruction of tooth structure through the development of small pits or cavities. The cause of dental caries is multifactorial, but begins typically due to the actions of bacteria, Streptococcus mutans. Individual factors may include structure of the tooth, immunological responses to bacteria, and composition of saliva in the mouth. It is currently understood that several different types of bacteria commonly found in the mouth, including Streptococcus mutans, colonize the surface of the tooth to form a substance called plaque. Plaque adheres to the teeth and is not removed will mineralize into tartar or calculus. Factors that contribute to the development of caries and plaque include frequency of eating, stickiness of the food, composition of saliva, presence of buffers, lack of access to preventive dental care, and overall oral hygiene. Dietary factors and eating patterns associated with dental health. Dietary factors associated with increased risk include sugar-sweetened liquids, sticky foods such as raisins, slow dissolving candies, sugary, starchy snacks such as cookies, cakes, simple sugars such as sucrose, honey, and molasses. Dietary factors associated with decreased risk. I'm going to say that again. Dietary factors associated with decreased risk include sugar-free chewing gum, mints and candies, fresh fruits and vegetables, high-quality protein foods, whole grain, low-sugar bread, and cereal. Caries are usually painless until they invade to the level of the tooth pulp. Eating patterns associated with increased risk include frequent and prolonged intake of food rich in simple sugars, eating sticky, retentive food alone, sipping sugar-sweetened beverages for prolonged periods, eating periods associated with decreased risk, space frequency of food and beverages intake at least 2 hours apart, select fresh, whole, unprocessed food to stimulate salivary output, choose sugarless gum for a brief period immediately after a meal or snack. It is uncommon for patients to suffer from inflammatory conditions of the mouth. When the mouth is inflamed or infected, maintaining oral intake is very difficult. Inflammatory conditions may result from poor dental hygiene or lack of dental care and can also develop in persons who are immunosuppressed or have undergone chemotherapy and radiation. Gingivitis is an inflammation of the gingiva. In gingivitis, the gums appear red and swollen and often bled. The tissue is very tender and painful. Other symptoms include fever, loss of appetite, foul breath, and a bad taste in the mouth. Prevention is key. Maintain good oral hygiene. Clean your teeth and mouth every 4 hours and at bedtime. Use frequent mouth rinses and avoid alcohol-based products. Treat oral lesions pharmacologically as appropriate. Antifungal medications if needed. Consider using an oral topical agent and an antiseptic such as vicos's lidocaine. Adjust textures and temperatures as tolerated. Extremes of very cold or hot are often not tolerated. Avoid carbonated beverages. General recommendations for nutrition therapy for jaw fractures and blenderized diets. Prepare all food to a very smooth and thin consistency. Cut food into small pieces and place in a strong blender or food processor. Commercially prepared baby food is available to use, but you will need to experiment with herbs and spices to add flavor. Adding extra protein, you can do that by adding dry milk powder, double strength milk, liquid egg substitutes. Blenderized meats, cottage cheese, yogurt, smooth blended, commercial liquid nutritional supplements for added protein, To add extra calories, you can use whole milk, half and half, evaporated milk and water in place of water as the fluid when it's blenderized. Add extra butter or margarine to blenderize vegetables, soups, or thin cereals. You can add gravy to the blenderized meats. Add sugar or honey to blenderize fruit, squash, pumpkin, or carrots. Dysgeusia is the condition of altered or impaired sense of taste. Asquesia is the inability to taste or mouth blindness. Both clinical conditions affect the ability of taste. For example, patients undergoing treatment for cancer relate changes in both taste and smell. Epithelial cells of the oral mucosa have a high turnover rate and can be affected by either chemotherapy or radiation therapy. Diseases of the tongue and palate can interfere with normal function of taste buds. The primary nutrition problem for diseases involving the oral cavity is the inability to maintain adequate oral intake. Foods may be difficult to swallow due to lack of saliva, thickened saliva, or the pain or of inflammatory condition or dental disease. When saliva production or quality is impaired, there's an additional risk of infection and dental caries. Common nutrition diagnoses associated with pathology of the oral cavity include inadequate oral intake, inadequate fluid intake, inadequate protein energy intake. Nutrition intervention strategies are focused on modification of distribution, type or amount of food provided. Texture modification of the current diet will be necessary for most of these clinical conditions. Monitoring and evaluation can be done through observation, a calorie count, or a food diary.